So to get back to our consideration of bacterial energy metabolism, now we have 1 hour and 10 minutes to go through fermentation pathways, different types of bacterial respiration, different types of lithotrophy, and photoautotrophy. That's our program. Now the objective is to understand some of these simple fermentation pathways. Well, simple in inverted commas, I guess. Now what you need to be able to do for each of these is to state what the end product is and cite some kind of genera of bacteria that use this particular fermentation pathway. So the first one, very simple. It's all going to start from pyruvate, okay? So if you remember glycolysis, start with glucose, somehow you metabolize it to pyruvate from one of the three glycolytic pathways. And the objective is to get rid of the excess NADH, because you need to regenerate NAD+ so that glyceraldehyde phosphate dehydrogenase can function and produce ATP, or rather get a free phosphate onto glyceraldehyde phosphate. Okay, so easiest way to do this, take the pyruvate, directly reduce it, and you get lactate. 3-carbon molecule, 3-carbon molecule. So it's very common, lactobacillus, streptococcus, lactococcus, bacteria that are used in making yogurt and other fermented products. The fermentation can be homo or heterolactic. So homolactic, you only get lactate. Okay. Heterolactic, you get lactate plus something else. Why might that occur? Well, it may depend on what kind of glycolytic pathway bacteria use. So if I just go back up a couple of slides here, okay, here was our summary of the different glycolytic pathways. So for Embden-Meyerhof and the Enter-Duderoff pathway, all you get is pyruvate. You get 2 copies down, 2 molecules of pyruvate down here from the Embden-Meyerhof pathway. From Enter-Duderoff, you get 1 kicked out a little bit earlier in the pathway, and then another one. So you just get pyruvate. Whereas with the pentose phosphate pathway, you get 1 molecule of pyruvate and 1 molecule of acetyl phosphate. So this will be, you remove the phosphate off of this, and this will be transformed into probably acetate, or metabolized in some other way. But you only have 1 molecule of pyruvate here, so this one could be transformed into lactate, and this will be transformed into something else. And that will give you a heterolactic fermentation. Okay, next one. So another possibility for fermentation is you start to go off with pyruvate again, kick off carbon dioxide. This gives you acetaldehyde, and then alcohol dehydrogenase will work on it to give you ethanol. And mostly this is yeast, right? Saccharomyces cerevisiae performs this one. But some bacteria do as well, so you should remember this one. Zymomonas performs ethanolic fermentation. Now these 2 fermentation pathways are very simple. So you might wonder, why do any bacterial species do anything else? Very simple, you get rid of the excess NADH, you regenerate NAD+, it works. What is the problem? Well, the problem is the accumulation of the end products. Ethanol, as everybody knows on Saturday morning, is a toxic molecule. Lactate, lactic acid, if this is produced in high quantities, the pH of the medium is going to go down. So the medium will become progressively more acidic. So the bacteria that produce lactic acid are going to have to be resistant. They'll have to be acidophiles if they're going to be able to grow, to continue to grow. That's, of course, the basis of the food conservation after the use of, well, heterolactic or homolactic fermentation. pH goes down, eventually all the bacterial growth stops, and no other bacteria can grow in this acidic medium. Ethanol as well, toxic. So these are the drawbacks, I say. So if bacteria can figure out another way to ferment and produce less of these toxic intermediates, either less acidic product or not produce ethanol but produce something less toxic, then that would be helpful. So one of the ways that this occurs is by butanediol fermentation. Okay, very common. Enterobacter serratia bacillus. Serratia is the one to remember for this. You start out with 2 molecules of lactate here. You've got 2 3-carbon molecules. They join together, so this is 5 carbon. CO2 is kicked out. Once again, CO2 is kicked out, so this becomes 4 carbon. And then in the last step, it's going to be reduced to 2,3-butanediol. So this is quite good because you don't produce so much of the toxic ethanol. You don't produce any acidic products here at all. So pH will stay neutral. This will probably have some unfortunate consequences on bacterial growth, but less toxic than ethanol. Now, this used to be a fermentation. This fermentation used to be important industrially because 2,3-butanediol was the starting compound for the synthesis of artificial rubber. So this was used at the beginning of the 20th century. However, it was replaced by petrochemistry. So it's not used anymore. And for that, it was serratia was used in that industrial process. So those 3, okay, so butanediol, lactic acid fermentation, ethanolic fermentation, these are the ones that are quite simple and relatively direct. For the other fermentation pathways that bacteria use, I'm going to have to take a bit of a detour around the normal uses of pyruvate in energy metabolism, in you or me or in Escherichia coli. Okay, so pyruvate is quite a central model, quite a central molecule in a lot of intermediary metabolism. So often this is going to be converted to acetyl coenzyme A. So this will be used in lipid fatty acid synthesis, pyruvate dehydrogenase, giving us even more NADH and kicking out carbon dioxide. So this becomes 2 carbon here. Pyruvate can also be carboxylated to oxaloacetate, and this is important for the synthesis of many amino acids. Let me just, I think glutamate, right? Oh, I didn't write it down and I've forgotten, okay? But important for one of the main pathways of amino acid synthesis. Starts with oxaloacetate. So bacteria need to do these 2 things anyway, okay? And in respiration, these are the 2 starting compounds of the tricarboxylic or Krebs cycle, which I'm just going to quickly summarize for you here. Okay, so you start off with acetyl coenzyme A, oxaloacetate, they give you citric acid, isocitric acid, alpha ketoglucaric acid, succinate, fumarate, malate, and then this turns around to oxaloacetate again. So this is 2 carbon, this is 4 carbon. Citrate, citric acid, 6 carbon, 6 carbon. You get carbon dioxide here, 5 carbon, get carbon dioxide here, and from here on in we're 4 carbon. So aerobic bacteria, and you generate a lot of NADH because the net product is what? What is happening? This 2 carbon, it's reduced, it's an acetyl group, so I guess it's CH3, it's like that. This is getting entirely oxidized to 2 molecules of carbon dioxide. That's the end product, and you get, I don't know, 3 or 4 molecules of NADH produced. Okay, that was a big problem for us if you were running fermentation. So when you have oxygen, that's not a problem, okay? So you can run around the Krebs cycle and you get rid of acetyl-CoA, you produce NADH and ATP. Now, if this same bacterium runs out of oxygen, okay, it's already got all of these enzymes. They're going round and round and round and round, but it's got no oxygen anymore. So this cycle is going to stop. And then how can it be used to perform fermentation? Well, as you may know, chemical reactions catalyzed by enzymes can run both ways depending on the substrate and the product and the concentration of substrates and products. So these steps, oxaloacetate, malate, fumarate, succinate, these are all Krebs cycle enzymes running in reverse. So instead of this cycle going around like this, going around this way. Once again, the starting compound is pyruvate, going to be carboxylated, and then these Krebs cycle enzymes are going to run it all the way to succinate, and this will use up 2 molecules of NADH. Instead of producing them, or rather reducing NAD plus into NADH, it's going to work the other way around. NADH will be oxidized. That's what we want to happen in fermentation. It solves the problem of the buildup of too much NADH. And at the end, the carbon dioxide is kicked out again, so you end up with a 3 carbon product, propionic acid. So 3 carbon, 3 carbon, but compared to lactic acid fermentation, you've got 1 acid group, 1 carboxylate group, but this molecule is more reduced. So you've managed to regenerate 2 molecules of NAD plus and not just 1. So in terms of how much NAD plus you regenerate for reduction in pH, this fermentation pathway is pretty efficient. More complicated than lactic acid fermentation because there are more enzymatic steps, but the bacterium already has all of these 4 enzymes, right? So it just needs to produce the enzyme that's going to catalyze the last step. It just needs 1 enzyme in addition to what it already has. So it's not so complicated for this fermentation pathway to evolve. Okay, and the last thing you need to know, propionic acid fermentation. This one's very easy to remember, Propionibacterium. Propionibacterium acnes is one that we all have, or many of us have. And last but by no means least are the mixed acid fermentations that many enterobacteria perform. So Escherichia coli does this one. Salmonella does this one. Okay, once again, we start from pyruvate, but it's not going to be used directly as the start of the fermentation process. It's going to be converted to acetyl coenzyme A. So normally, during respiration, this step will be catalyzed by pyruvate dehydrogenase. But that's bad for fermentation because it gives us one more molecule of reduced NADH. So for a mixed acid fermentation, the first step is not catalyzed by pyruvate dehydrogenase, but by pyruvate formate lyase. So in this case, the two products of the reaction are formic acid and acetyl coenzyme A. So this is one carbon, right? This is two carbon. Oh, I just forgot something I should say here. Right then, go back and look at the requirements of bacteria for growth factors. One of these reactions requires thiamine, and one of them requires biotin. Which one and why? Remember what I said about thiamine and biotin? Why they are essential? B1 and B7. Essential enzyme cofactors. Thiamine, 2 carbon transfers. Biotin, 1 carbon transfers. 2 carbon here. Going to be transferred from pyruvate, kick out the CO2, so the 2 carbon is transferred onto coenzyme A. So this will be a, I guess, thiamine dependent reaction, and this one biotin dependent. But okay, let me go back to what I should be talking about in the first place here. Okay, so instead of producing acetyl-CoA through pyruvate decarboxylase, Escherichia coli will first produce formic acid plus acetyl-CoA. And then this acetyl coenzyme A will be used for a whole bunch of different things. Now, this is almost impossible to read, right, on the projected slide. Butyrate, butanol, propanol, I guess, and acetate. Why are they doing all this complicated stuff? The only thing you want, the basic function of fermentation is regenerate NAD+, so you could keep glycolysis turning around and produce ATP. Now, the advantage of this complicated fermentation pathway is some of these steps allow the bacteria to regenerate extra copies or extra molecules of ATP. So this is complicated, but it's complicated for a reason, because it allows the bacteria to squeeze out a couple of molecules of ATP here and there. So that's the main advantage. What is the disadvantage? Well, one of them is production of a large amount of formic acid. So once again, it's like with lactic acid fermentation, the pH will go down. So as the bacteria use up the glucose, formic acid will be produced, the pH will go down. Now, E. coli can get around this a little bit. As the pH goes down, they produce another enzyme which will cleave formic acid into hydrogen and so dihydrogen and carbon dioxide. So that means that when E. coli is grown at neutral or slightly alkaline pH, they don't need this enzyme. They produce a lot of formic acid. So this is one of the major fermentation products. As the pH goes down, they'll produce this enzyme and they'll start producing carbon dioxide and hydrogen. And is there anything more to say about this? So this is going on in our guts all the time. And one of the interesting things is that this part here, acetyl-CoA, is going to be reduced into acetaldehyde and then ethanol. So your gut bacteria are producing small amounts of ethanol as a part of your normal life. So that means it's impossible to have a zero value for an ethanol test when you're driving, because this ethanol is produced in your gut from fermentation of whatever you eat, the sugars that you eat, and some bacteria will produce a small amount of ethanol. It will be absorbed into your bloodstream. So the amount of ethanol that you have circulating in your blood is not really zero, even if you haven't drunk any. And there are some people who've got a kind of weird syndrome. They can get drunk, but without drinking any alcohol. And it's because they have something going on with their gut bacteria that produce a lot of ethanol by fermentation. So there's some people, they've got, I think it's also been related to if they have a lot of sugary food or something like that, and then later on, and it occurred because someone was caught driving in a strange way. And I think it was a lady, she was tested for blood alcohol, and she had an illegal blood alcohol limit. And she said, yeah, but what, I haven't been drinking. Believe me, officer. And of course, the policeman said, yeah, I've heard that one before. But in fact, she was telling the truth, and she had a lot of medical tests to prove that, in fact, she had a blood alcohol level that was abnormally high. And it was to do with her gut bacteria. Okay, so, no, anyway, I'm not going to incite anyone to drink and drive. It's a very dangerous thing to do. And if you ever get caught, you should own up to it and take your punishment like a man or woman. Okay, so let's have a little break, and I'm going to ask you these questions. You want to do this by clickers or just a show of hands? Just a show of hands would be okay, right? Yeah, I think so. Everybody okay? So, limitations of fermentation as a form of energy metabolism. Energy yield per carbon is low. Who says yes? Requires oxygen. Who says yes? Or another electron acceptor. 2 or 3 people. Only used by gram-positives. Nobody. End products can be toxic. Oh yeah, almost everybody. Requires cytochromes. Nobody. Yeah, I never talked about cytochromes, so can't be right. Mostly all right, but the first one, energy yield per carbon is low. This is also a limitation. You need a lot of sugar hanging around or organic carbon. And it doesn't all get used up, okay? So all of the fermentation products that can be toxic, that's correct. If oxygen is present, these can be used by other organisms, okay? We could eat yogurt and we get the energy value from the lactic acid, because we have oxidative energy metabolism. So energy yield per carbon is low, and the fermentation products still retain, quite a lot of organic carbon that can be used by other organisms. We can get energy from them. Okay, so up to this point, summary is, okay, we did this last time, chemotroph, phototroph, organotroph, lithotroph. Don't confuse auxotroph, autotroph. And the key points about fermentation are here. Problem with glycolysis is that there's too much reduced NADH or NADPH. So any kind of fermentation pathway needs an electron acceptor, and it's always pyruvate or some kind of metabolic intermediate derived from pyruvate is always the electron acceptor. It only occurs in the absence of oxygen. Okay, so there's no other electron acceptor around, just the pyruvate that's derived from the original carbon source. And some of the fermentation products are valuable. Okay, lactic acid fermentations for food conservation, ethanolic fermentation, mostly yeast for alcoholic beverages. Butanediol used to be an important industrial process, not so much anymore. Okay, so that's anaerobic energy metabolism by fermentation. Anaerobic chemoorganoheterotrophs. However, what happens if you have oxygen available? Well, the first part is the same. You start off with glucose, you start off with glycolysis, you get pyruvate. Then what happens is bacteria will run the Krebs cycle, illustrated here. And the overall result is that all of the carbon is going to be oxidized to CO2. And this produces a lot of NADH. However, that's not a problem because this is going to, the electrons on the NADH are going to be transferred onto oxygen as the terminal electron acceptor. So these electrons get passed down an electron transport chain and produce oxygen. So these are the main steps. Glycolysis to produce pyruvate acetyl-CoA. Krebs cycle oxidizes the carbon to CO2. That gives you a lot of NADH. And then these electrons are transported onto oxygen through the electron transport chain. And finally, this allows even more ATP to be generated. So what you need to understand is how electron transport can be coupled to ATP generation. So in E.coli, there's a simple electron transport chain with 3 components. NADH dehydrogenase, quinones in the membrane, and cytochrome oxidase. So what are each of these things doing? So NAD dehydrogenase takes the reduced form, NADH, and takes away its electrons and leaves NAD+. So these electrons go on to, you don't really need to know this, flavine adenine dinucleotide and an iron sulfur complex. The main point is they're going to be transferred onto quinone. So this is a small organic molecule, lipid soluble, so it's inside the membrane. So quinone picks up 2 electrons that come from NADH. So it's got a negative charge. Okay? To balance out the negative charge, it picks up 2 protons from the cytoplasmic face of the membrane, and this gives you reduced quinone, QH2 here. QH2 gives up its electrons for the cytochrome complex, and the protons are released on the periplasmic face. This is very important because what has happened is the protons have been picked up and the cytosol and released in the periplasm. So the quinone cycle allows a proton gradient to build up. And in the last step, these electrons that have been passed to the cytochrome complex from quinone are transferred onto molecular oxygen, together with some more protons to produce water. Now in E. coli, there are 2 cytochrome complexes. There's the BO branch. If you've got high oxygen conditions, E. coli will use BO because this complex can also transfer protons across the membrane. So you get a bigger proton gradient. However, the affinity for oxygen is low. So it only works when oxygen is in excess. If oxygen is limiting, then it's the BD branch that will be expressed. Okay, so the main point is through the quinone cycle and through the BO cytochrome, protons are translocated across the membrane. So you get a proton gradient, proton motive force. And then this is used to produce or to fuel ATP synthesis by the proton ATPase. So from each NADH, you get a bunch of protons that are translocated. Apparently, you need 3 to produce 1 molecule of ATP. So once all the numbers are added up, the theoretical maximum for aerobic respiration is a yield of 38 ATPs per glucose. So this is much more efficient than fermentation, where we were talking about 1 or 2 molecules of ATP per glucose, depending on the glycolytic pathway. Okay, so whenever you think this is too complicated, all of this Krebs cycle, electron transport, all this stuff is very complicated. You need a lot of proteins to do all of this. Why? Why do they bother? Well, they bother because they get a big payoff in terms of energy, in terms of ATP. So that makes it all worthwhile, okay? Anaerobic respiration. This sounds like it should not exist, right? Anaerobic means without air. Respiration, normally something to do with air. But in biochemical terms, respiration is just transferring electrons from glucose onto some kind of inorganic electron acceptor. So for aerobic respiration, the electron acceptor is oxygen, whereas in anaerobic respiration, then it's some other inorganic electron acceptor that can be reduced. So often it's nitrate, which can be reduced firstly to nitrite or then to nitrogen gas. I don't think you really need to know the details about this. The thing that's important, okay, so this is for Paracoccus denitrificans. So it's got an electron transport chain. It starts off the same as E. coli, okay, so you've got an NADH dehydrogenase. You're going to take the electrons off. They're going to be transported onto coenzyme Q. And once again, this is going to cycle around the membrane, produce a proton gradient. And then it's the end part that's different. It's the cytochromes. They're not going to transfer the electrons onto molecular oxygen. They're going to transfer them onto, whoa, what's going on here? Nitrate. Yeah, okay. Oh yeah, this is the oxidative form. So here we go. The NADH dehydrogenase, coenzyme Q, but then the electrons get transferred onto nitrate to give you nitrite, and then onto nitrite to give you, I know, nitric oxide, nitrous oxide, and then nitrogen gas. Why does it work? It's because the redox potentials for these half reactions make nitrate a good electron acceptor. So your electron donor, NADH, is up here. This is a pretty big change in redox potential. So it's highly favorable. So nitrate is not the only electron acceptor that can be used in anaerobic respiration. So in well, in bedrock, when you have groundwater flowing through rock. And if you have organic carbon, then different populations of bacteria will use different, well, bedrock, maybe not bedrock, well, let's say, yeah, well, I think so. Okay, but different populations of bacteria will use different electron acceptors to carry on growing. So first, oxygen is the best one, okay? Then nitrogen, or nitrate, is the second best one, so this will be used up secondly. And apparently manganese is the next best one, then iron, then sulfur, and then finally, when you get right down to the base, there's none of these oxidized good electron acceptors are left at all. The only thing that's left is carbon dioxide, which can be used as an electron acceptor by methanogens. So you get methane produced underground by bacteria. Okay, but you have to have organic carbon coming in somewhere. Okay, at the beginning is organic carbon, a fermentation pathway, and then you have a buildup of NADH. You have to, and then the bacteria have to find some kind of inorganic electron acceptor. Now, chemolithotrophs, they're almost doing the exact opposite of these anaerobic respiratory bacteria. Okay, so these guys, they start off with organic carbon, they get the electrons from the organic carbon, and they put it onto some kind of inorganic electron acceptor. Chemolithotrophs, they're doing the opposite. They're using an inorganic electron donor. They use that to generate metabolic energy, and they use that energy to fix carbon dioxide and reduce it to produce sugars, fatty acids, lipids, everything they need. So, in order for this to work, then bacteria need some kind of reduced inorganic chemical. And they're going to base their metabolism on electron transfer from an inorganic donor to an inorganic acceptor. And we're going to look at how hydrogen oxidizers, nitrifying bacteria, and sulfur oxidizers get their metabolic energy. So the first one, the simplest one, perhaps, is hydrogen oxidizers. So if you start with molecular hydrogen, this can give up, or let's just imagine that it could possibly be separated into 2 protons and 2 electrons. This is apparently very favorable. Molecular hydrogen is a good electron donor, so much so that its redox potential is minus 0.42 volts. Even a better electron donor than NADH. So what does that mean? That means that NADH can be produced directly. Yeah, why did I go over here? I wanted to go over here because okay, so if you remember the 3 forms of energy, metabolic energy that bacteria need, okay, reducing equivalents, you need to reduce carbon to make, these guys need to fix carbon, so they need reducing equivalents, NADH. Proton motive force for moving about, symport, antiport, and ATP for a whole bunch of other biochemical reactions. So in the case of hydrogen oxidizers, okay, and it's simple. You've got hydrogen can give you NADH directly because the redox potential is good enough for the electrons to be transferred directly onto NAD+. Then this can be used to produce proton motive force. You have an electron transport chain, transport protons across the other side of the membrane, and then that can be used to give you ATP. Okay? So this is the way things work. It goes this way, and that way, then that way. Simple, right? Okay, so the kind of bacteria that use hydrogen oxidation are bacteria that live in hot volcanic springs where molecular hydrogen is released as the gas is released from the springs. So hydrogen is available from geothermal sources, and these are on the surface of the Earth, right? So oxygen is available as the electron acceptor. So then this is a very, very favorable reaction. You know, if you do this in a chemical tube, it will explode. So it's going to release a lot of energy. So that's how these guys live. The other bacteria that do this are methanogens. Now they can use molecular hydrogen as their source of, as the electron donor, carbon dioxide as the electron acceptor. Now where do you find these two things together? What does Escherichia coli do? It's fermenting away, the pH starts to go down. The bacteria think, I'm not happy, it's too acidic for me. I must stop producing this formic acid. What can I do with it? Yeah, they cleave the formic acid into these two things. So inside your guts, then, well, many mammals, not necessarily all of them, but many mammals have methanogens that live in their guts, and they use the hydrogen, the carbon dioxide that are kicked out by E. coli as a product of their fermentation and produce methane. Cows, right? They produce a lot of methane. Yes. They don't have any organic carbon. That's what's missing. They have an inorganic source of electrons. They have an electron acceptor that's very good, so they have a very favorable chemical reaction, but they have no organic carbon. So these are going to be autolithotrophs. Actually, that's true for all of these lithotrophs. They manage to scratch out a living from these inorganic reactions that are mostly give a pretty poor energy yield. This is the best one. If you have molecular hydrogen, it's great. You know, go and use it. But if you don't, things get really tough, and they have to try and use a chemical reaction to get NADH so they can fix carbon dioxide and reduce it and make sugar. Okay, nitrifying bacteria. Here they're going to use reduced forms of nitrogen, ammonia or nitrite, and oxidize them into nitrate or nitrite with molecular oxygen. So you should know these bacteria, okay? So Nitrosomonas and Nitrobacter. I was thinking about how to remember this just earlier. So Nitrosomonas sounds like ammonium, right? Ammonium, ammonas. Okay, so Nitrosomonas will use ammonium and oxidize it to nitrite. Nitrobacter oxidizes the nitrate back to nitrate. Okay, well, anyway, I think it helps me remember it anyway. So when you look at the half reactions here, nitrite is supposed to be the electron donor. Okay, but if you look at the redox potential, it's not so good. Okay. Rather, nitrate is a pretty good electron acceptor. But, of course, where's our thing here? Okay, nitrite is up here. Oxygen is even better as an electron acceptor. So nitrite can be an electron donor if the electrons are going to go to oxygen at the end, because you still have the gap here. However, you can't directly transfer these electrons onto NADH because the redox potentials are the wrong way round. So what is going to have to happen here? You're going to have nitrite as the electron donor. Oxygen is the electron acceptor. Okay, these electrons are going to go. Somehow, during this electron transport, then you have to generate proton motive force. Maybe it's a quinone cycle and you get the protons transported across the membrane. And then you can use this to push electrons back onto NADH. And you also use it to generate ATP. Now, how can that possibly work? If you go back to what I tried to explain in the concepts of, how can a proton gradient be related to a redox potential? What is important is the difference between the concentrations on the side of the membrane, right? If you remember our old friend, the Nernst equation, this is what's different. So if you have a big enough gradient, you can push an unfavorable redox reaction to go in the reaction you want. So that's how this metabolism is working. Even though it should be impossible, okay? What it means is that you're going to have to use a lot of this nitrate to nitrite in order to generate even one NADH, because you can't do it directly. And the way that you could kind of build up the force and build up the chemical potential is by building up the proton motive force. And the last one are sulfur oxidizers. So, yeah, where do you get these guys, nitrifying bacteria? You get them in the soil. Ammonia, this kind of stuff. This is an excretion product for you and I. Animals will excrete ammonia in the form of urea. Then it will be degraded to ammonia. And then nitrifying bacteria can regenerate it to nitrate. And the plants can take it up. Sulfur oxidizers. So these guys are getting by with oxidation of reduced sulfur compounds, hydrogen sulfide or molecular sulfur. Now, hydrogen sulfide is a pretty good electron donor. So either molecular oxygen or nitrate can be used as the electron acceptors. What else to say about it? Okay, this part, I include this part because it's a quite unusual way to generate ATP. So some sulfur oxidizers can incorporate sulfite into this molecule, which is adenosine phosphosulfite. And then, during the cleavage of the phosphosulfite bond, then sulfate can be released, and this gives you 2 copies of ADP, which can combine together and give you 1 molecule of ATP. So this is an unusual way of generating ATP that doesn't depend on proton motive force or an electron transport chain. Last thing to say about this is, where do you find these guys? Once again, Thiobacillus denitrificans. Maybe you can find this one in hot springs where you have hydrogen sulfide. Not necessarily in an aerobic environment. In the absence of oxygen, this will also work. Thiobacillus ferrooxidans is wherever you have a large amount of iron sulfide in old mine workings. And this can be an important problem. So in the States, in the Appalachian Mountains, there are a lot of old mine workings that are left open. So you have iron 2, sulfide in contact with oxygen. These bacteria can grow in the mines in the dark. And it's resulted in the release of millions of tons of sulfuric acid into the rivers. So it's had an important impact on the environment, these bacteria. Now, fortunately for us, people understand the basic biochemistry of what these bacteria are doing, and everybody knows that it's necessary for these bacteria to have access to oxygen, otherwise their metabolism can't work. So that gives a very simple solution to the problem. Just block up the mines so that there's no access to the outside air, and then this metabolism stops. Okay. So let's see if you understand all of this stuff about anaerobic respiration and chemolithotrophy. Oh, okay, let's go. So how about this one? Glucose as an electron donor, nitrate as an electron acceptor. Can work? 2 or 3 people say yes. Glucose as an electron donor, CO2 as the electron acceptor. 1 person, 2 people. Nitrite as an electron donor. So some of them, it's not just one correct answer here. Okay. Nitrite as an electron donor, CO2 as the electron acceptor. 1 person. Ammonium as the electron donor, sulfate as the electron acceptor. Nobody says that. Molecular hydrogen as the electron donor, nitrate as the electron acceptor. 1 or 2 people. Most people are not putting their hands up. So we've got like 3 or 4 people. So most people have not understood this. Okay. This is what you need to be able to answer this question. Okay, you need to have the redox potentials of the different half reactions so that you can understand which compound is a good electron donor and which compound is a good electron acceptor. And when I say good, it only has to be good enough, okay? So what was the first one we had? We had glucose as the electron donor, nitrate as the electron acceptor. So glucose is going to be oxidized to CO2. It's a very good electron donor up here. It's minus 0.5 electron volts. Nitrate to nitrite, this is the electron acceptor. This is very favorable. This is glucose, a very good electron donor. Nitrate, pretty good electron acceptor. Okay, so this will work. Number 1, the first one, yes. That's what happens in anaerobic respiration. Glucose is the electron donor. Nitrate is the electron acceptor. nitrate-dependent anaerobic respiration. It's what Pyrococcus denitrificans does. Okay, the next one, glucose as the electron donor, CO2 as the electron acceptor. I would say that's wrong, right? So it's glucose as the electron donor and oxygen as the electron acceptor. So probably wrong. Although you might think, yeah, but if you ferment the glucose and you produce hydrogen, then you can use the carbon dioxide as the electron acceptor. Convoluted, but I think this is basically not supposed to be that complicated. Okay, probably wrong for this one. How about this one? Nitrite as the electron donor, CO2 as the electron acceptor. Also is not going to work. Molecular hydrogen as the electron donor, nitrate as the electron acceptor. Is that going to work? Molecular hydrogen is up here, very, very good electron donor. Nitrate is the electron acceptor. Will also work. Okay, it's good. And the last one, ammonium is the electron donor and sulfate is the electron acceptor. Can that possibly work? They're not on here. But it doesn't work. Okay, it works the other way around. Okay, because reduced sulfur is a better electron donor than reduced nitrogen compounds. Okay, so these nitrogen oxidizers, they all need molecular oxygen. Nitrogen, nitrate, ammonium. Yes? Yes. But it has a negative value. Okay, if you have if it's minus something volts, that means this is a good electron donor. If it's plus some kind of volts, it's a good electron acceptor. So all of these scales, it's the negative values at the top and the positive values are at the bottom. The higher up on the scale, the better the electron donor is. Okay, so that is why some bacteria can live as chemolithotrophs, because they have a good inorganic electron donor and some kind of electron acceptor that's nearby. So they can use this favorable inorganic chemical reaction to get some kind of metabolic energy. But they don't always work in the same direction. The big difference is whether bacteria have oxygen present or not. Okay, because if oxygen is present, there are a lot of inorganic chemicals that can be used as electron donors. And then chemolithotrophs can live. If oxygen is absent and you have organic carbon, then what's going to happen is the opposite, anaerobic respiration. The inorganic molecules are going to be used as the electron acceptor. Okay, so think about this. Ask yourself, do I have oxygen present? Yes, no, what is going to be possible? Because this is the big thing that determines what kind of bacterial metabolism is going to occur. Respiration, chemolithotrophy, they'll work with oxygen present. Fermentation, anaerobic respiration, oxygen is absent. Okay, we just have 10 minutes for phototrophs, and yeah, I'm just going to go over this very quickly, and there are maybe 2 or 3 points that you will need to remember. Now, the first one is that when you look at the distribution of photosynthetic bacteria in the phylogenetic tree, there are a bunch of different photosynthetic bacteria, and they're not all in one phylum. Okay? So within the eubacteria, they are kind of spread out through gram-positives and different groups of gram-negatives, as well as Chloroflexus, which is one of these very basal groups. And there are also some photosynthetic archaebacteria, halobacteria. So the light harvesting complexes are very, very different in archaebacteria and eubacteria. So that means what? Two things. These are totally different systems. They occurred independently. So you have two origins of photosynthesis in eubacteria and archaebacteria. However, all of the eubacterial photosynthetic systems are based on bacterial chlorophyll. So this indicates a common origin. But all the photosynthetic bacteria are everywhere in the phylogenetic tree. So how did that happen? Well, lateral gene transfer, the thing that mixes up and causes problems in bacterial phylogeny. So somewhere, some kind of ancestral bacterium developed photosynthesis, and then it was transferred to the other groups, photosynthetic bacteria. And, okay. So that's a scheme for that, but I'm not going to go into it. Okay, that's the first point. The second point is, what is the photosynthetic metabolism doing? So in archaebacteria, Bacteriorhodopsin, it works as a kind of proton pump. So protons that are in the cytosol get, transported through here. Light allows them to be transported out to the periplasm. So what does that mean? That means if these guys have got light, then they can produce proton motive force. No electron donor, no electron acceptor. Now, these guys, they need to get organic carbon from somewhere. Otherwise, they're heterotrophs. So they depend on organic carbon for their for their source of carbon and for electrons. So why don't they just use the organic carbon and respire? Well, when they grow in low oxygen conditions, they produce bacteriorhodopsin, and this allows them to get more energy than if they were just doing fermentation, performing fermentation. So they get a bit of a plus. And that's also the same thing that purple sulfur, non-sulfur bacteria are doing. Okay? They only have a type 2 photosystem. What is happening is the photosystem will be activated by light, and then the electrons are going to be transferred onto quinone. Quinone again. So this is going to cycle around in the membrane and allow proton transfer from one side of the membrane to the other. So it's doing the same thing as in the halobacterium, the archaebacteria. So once again, these purple non-sulfur bacteria, if they have carbon, if they have organic carbon and oxygen, they'll grow as heterotrophs. If they have low oxygen conditions but light, then they will grow, they will express their photosystem too, and use this to get ATP. And the last one are green sulfur bacteria. They only have photosystem 1. And in this case, they have an inorganic electron donor. Why do they bother to photosynthesize? We saw that that lithoautotrophs can use sulfide to transfer the electrons onto molecular oxygen, and that will work. Now, if they boost up the redox potential of this electron transfer from the light, then that means they can generate NADH directly. So it's more efficient. And they also get proton motive force, and that can be used exclusively for ATP generation. Okay, I'm going to go on to the last slide, because I can see by the looks in your eyes, it's all glazing over. That's enough bacterial metabolism, I can tell. Okay, so all of these different photosynthetic bacteria, apart from the cyanobacteria, these guys are the real specialists, okay? That's how they live, it's their thing. This was probably the first group that managed to get photosynthesis to work in eubacteria. And then they transferred for either photosystem 1 or photosystem 2 to these other groups of bacteria. All of these other groups of bacteria were already living and doing something else for their energy metabolism. So the non-sulfur bacteria, purple and green non-sulfur bacteria, were basically chemoheterotrophs. They used organic carbon in a kind of low oxygen environment, and becoming photosynthetic allows them to squeeze out a little bit more energy from the organic carbon that they have. Purple sulfur bacteria and the green sulfur bacteria, their basic metabolism is they're kind of lithoautotrophs, okay? So they're scratching a life out just from oxidizing reduced sulfur compounds. If you add a bit of photosynthesis on top of that, it becomes more efficient. So they get more energy. Okay, so this is the summary for today's and last week's lecture. Okay, respiration. Well, how's it working? You start out with reduced organic carbon. This allows you to generate NADH, which is used to produce. Okay, via an electron transport chain, you get proton-motive force, and that gives you ATP. Very complicated. You have to understand why. What is the point of all this complication? You also need to understand, very important, why anaerobic respiration is not the same thing as fermentation. Chemolithotrophs. Here, the first thing that's going to happen is electron transport. They're getting all their energy from a redox reaction. So that's the first thing. And then this can be used either to generate proton-motive force and then ATP, NADH directly or indirectly. So for hydrogen oxidizers, NADH will be generated directly. For all the others, you're going to have to generate proton-motive force and use that to generate NADH. Why do you need it? You need it to fix carbon and reduce carbon dioxide to organic carbon. And this only works with particular donor-acceptor couples. And that depends on the redox potential. Okay, that's it for today. So I will send out another microbe of the week tonight, 8 p.m. Okay, and I will try to answer this time. Thank you.